Hello, it's my pleasure to welcome you to this virtual scientific meeting developed by the American Neurogastroenterology and Motility Society, focusing on basic concepts in neurogastroenterology. My name is Beverly Greenwood van Meerveld, and I'm the immediate past president of the ANMS and current director of the ANMS Institute. The title of today's ANMS Symposia is Emerging Concepts and Technologies for Researchers in Neurogastroenterology. We have three excellent speakers, and on behalf of the ANMS, I would like to thank each of you for your presentations tonight. Before we start, I'd like to share with you a few important dates. As a reminder, the 2022 ANMS clinical course is in Philadelphia on August 5th to 7th. I'd also like you to save the date. In 2023, we have the joint ANMS clinical course, scientific meeting, and young investigator forum. This will be in Austin, Texas on August the 11th through 13th in 2023. So please visit the ANMS website for details of all ANMS events. It's now my pleasure to introduce today's moderators, Dr. Nicholas Verne and Dr. Andrea Harrington. Dr. Verne is professor of medicine at the University of Tennessee College of Medicine in Memphis, Tennessee. His research focuses on the neurobiological mechanisms of altered motility and visceral nociception in functional gastrointestinal disorders. Over the last decade, Dr. Verne's laboratory has examined the aberrant non-coding RNA signaling pathways in IBS patients and in animal models of visceral nociception. In addition, his group has studied the neurobiological basis of GI disorders in veterans with Gulf War illness. And his current research focuses on the use of small molecules, specifically miRNA mimetics and inhibitors as targeted therapies for IBS patients. We are also honored tonight to welcome Dr. Andrea Harrington to co-moderate tonight's event. Dr. Harrington is a senior research fellow in the Visceral Pain Research Group at Flinders University in Australia. And her research is aimed at unraveling the spinal cord and brain circuits that receive and process sensory information from the viscera. And she is investigating remodeling within these circuits in various models of chronic visceral pain. Her technical strengths are in vitro and in vivo neuroanatomical mapping, molecular localization, microscopy, and neurophysiology approaches. Thank you both for co-moderating tonight's event. I'll turn over the program to Dr. Verne.
Great, thank you, Beverly. Well, welcome everybody to this evening session. Before I introduce the first speaker, I'd like to remind you to please put your questions in the question and answer box. We are going to have a Q&A session at the end after our third speaker has finished. Well, with that, it's a great pleasure to introduce our first speaker, Tamas Ordog, who is professor of physiology and director of the gastroenterology research unit at the Division of Gastroenterology Hepatology at the Mayo Clinic at Rochester, Minnesota. He is also genetics and epigenetics mechanistic research theme group leader and epigenomics core director in the Center for Cell Signaling and Gastroenterology. His research focuses on the transcriptional, epigenetic, and metabolic regulation of gene expression in the lineage of interstitial cells that Cajal, gastrointestinal stromal tumors, and enteric neurons. He's contributed to the discovery of mechanisms of epigenetic inheritance at mitosis enduring stem cell differentiation and the role of epigenetic factors in diabetes, aging, immunologic, fibrotic, psychiatric, and neurodegenerative diseases, as well as various cancers. His research interests also revolve around molecular, cellular, and translational biology of the gastrointestinal neuromuscular system, which with particular focus on the gut's own nervous system and interstitial cells at Cajal, which mediate interactions between nerve and smooth muscle cells and generate electrical rhythm required for gut peristalsis. Deletion or loss of function of subsets of gastrointestinal neurons and interstitial cells of Cajal, which leads to gastrointestinal motility disorders, for example, as we see in diabetes, whereas neoplastic growth of interstitial cells at Cajal underlies gastrointestinal stromal tumors, the most common form of sarcoma. We're pleased to welcome him this evening. The title of his talk will be Epigenomics.
Good evening, everybody. Thank you very much, Nick, for the very kind introduction. I'd like to thank also to Andrea, Lori, and Beverly for the invitation to speak about epigenomic concepts and technologies in this forum. I have one disclosure to make. Perhaps the easiest way to consider or understand epigenetics is to consider this a study of mechanisms that enable the expression of multiple distinct cell type and developmental stage-specific phenotypes from a single genome. Epigenetic functions are not restricted to the control of gene expression. In fact, epigenetic mechanisms regulate all templated functions in the genome. They include DNA replication and DNA repair as well. Since these are recursive mechanisms, it is fair to say that epigenetic mechanisms can be considered gatekeepers and guardians of genomic information. Epigenetic functions regulate templated mechanisms through multiple ways, by regulating accessibility of the template, by recruiting regulatory complexes, and by controlling folding of the genome to bring in proximity regulatory elements that may be separated by large genomic distances. Epigenetic mechanisms also control protein synthesis through regulating RNA splicing and RNA stability. In the interest of time today, I'm going to focus on the regulation of RNA transcription, since most investigators study this aspect of epigenetics. Central to the concept of epigenetics is a set of covalent marks that exist on the DNA template or the chromatin template, which form some kind of a combinatorial code that exists above and beyond the genetic code. This combinatorial code defines so-called epigenetic states. Epigenetic states can be classified broadly into active and repressive states. Repressed chromatin marks are actually acquired during mitosis through epigenetic inheritance, semi-conservative replication of the chromatin onto the daughter cells, which is actually tightly linked to DNA replication. In addition, sequence-specific transcription factors can recruit code writers, which then establish de novo repressive regions in the genome. The same concept also applies to activating mechanisms. Transcription factors can recruit these code writers. But we also must consider a very important recent discovery, or actually the proof was acquired recently. This has been suspected for quite some time, that the process of transcription itself is critical for establishment of active chromatin regions. Now, both sets of transcription factors also recruit code erasers to control usually the opposing marks. Together, these mechanisms lead to the establishment of epigenetic states, which are then read and interpreted by code readers, which are part of effector complexes that also execute the epigenomic functions. You all know a lot about letters of the epigenetic code already. I just briefly want to mention that these consist of DNA and RNA bases that are modified without changing the code. A notable and well-established, well-known modification is the methylation of cytosine bases. In the case of histones, the modifications actually decorate amino acids that are sprinkled throughout the primary sequence of the core histone proteins, H3, H4, H2A, and H2B, which together form the structure of the nucleosome. Now, most of these modifications actually reside on the unstructured tails and C-terminal tails of these nucleosomes, which makes them rather accessible. If you want to measure these epigenetic code forming modifications, we have to consider the uncertainty principle of epigenetics, namely that if you want to accurately measure these modifications or any other epigenetic variables, you usually have to use some form of liquid chromatography tandem mass spectrometry, which does not permit the analysis of genomic localization of these marks. Therefore, we have to use separate methods to assess the genomic localization of these marks or more intramolecular localization of these marks, but these methods are unfortunately not that quantitative. Nevertheless, we can still make quantitative conclusions or semi-quantitative conclusions, and there are mitigation techniques available that make genome-wide epigenetic techniques more or less quantitative. These techniques that assess the genome-wide distribution of epigenetic marks are listed in this table. In the case of DNA modifications, hybridization is still commonly used in connection with microarrays. Chemical derivatization, specifically bisulfite modification combined with next-generation sequencing, is a frequent technique to assess DNA methylation, hydroxymethylation. Direct sequencing using the Oxford Nanopore system is less often used, but it is emerging as an important tool to assess RNA modifications. Now, all of these modifications, DNA, RNA, and histone modifications, as well as the assessment of chromatin regulators, is possible using antibody-based techniques, which are combined with immunoprecipitation, next-generation sequencing. We call these techniques, depending on what we are targeting, DNA, RNA, and chromatin immunoprecipitation. This is a core method, so it is important to say a couple of words about this method. It is typically applied to fixed cells from which chromatin is isolated, and then the chromatin is sheared either mechanically or digested enzymatically, or the combination thereof, which we actually prefer. Then antibodies are used to detect the target and modification or a chromatin-associated protein or a transcription factor, and we perform immunoprecipitation, and then we liberate DNA from these complexes and interrogate it usually by next-generation sequencing, where the targeted methods are also available. This technique is quite wasteful. It requires a lot of input, and it is quite difficult to perform. Therefore, laboratories came up with some clever ways to simplify the process and improve its sensitivity. Specifically, the cleavage on the targets and release using nuclease or cut and run technique. That was the first one that was developed by the Henikoff lab. That has become quite popular over the past couple of years. It involves a protein A micrococcal nuclease fusion protein, which is reacted with the antibody that detects the target. The micrococcal nuclease cleaves the DNA surrounding the target, enabling the separation of this complex and more efficacious identification and analysis of the associated DNA. Another method that changed the field involves Illumina's TM5 hyperactive transposase, which is a mutant transposase that actually acts as an endonuclease and can affix a payload, a piece of DNA, in this case, a sequencing barcodes to the cut ends. This way, one can generate libraries, sequencing libraries in a single step. The Henikoff group applied this technique, also applied this technique to the cleavage on the targets method, developing this cut and tag technique that replaces the protein A MMA's fusion protein with protein A TM5 transposase fusion protein. Here, the TM5 will cleave the DNA around the target and at the same time barcode the fragments for one-step library preparation. In addition to the histone marks, the position of the nucleosomes also carries important epigenetic information. This is because there are these so-called nucleosome-free regions that are devoid of nucleosomes, as the name implies. And these regions can be found in regions, genomic regions that are either active or poised for activation. Therefore, they are functionally important. So one way to interrogate nucleosome positions is to perform micronuclease sequencing. Micronuclease can cleave the DNA, as I mentioned earlier, but only outside the nucleosomes. Isolating the nucleosome-associated DNA and sequencing this DNA enables the mapping of nucleosome positions throughout the genome and identifying these nucleosome-free regions. The problem with this technique is that it is quite expensive, requires very deep sequencing, since there are 30 million nucleosomes per human cell. Therefore, investigators developed alternative ways to assess open chromatin regions. There are several methods, but they have all been superseded recently by ATAC-seq, which stands for Analysis of Transposase Accessible Chromatin by Sequencing. It utilizes the same hyperactive transposase, which can cleave and can only cleave a nucleosome-free DNA and decorate it with these barcodes, sequencing barcodes, and these barcodes enable the direct sequencing of the fragments after isolation. This method has been shown to be very good for detecting regulatory elements, active and poised regulatory elements in various genomes, identifying cell types, identifying transcription factors bound to specific sites, and predicting regulatory element gene interactions. Very importantly, this method has also enabled the prediction of the functional significance of genetic variants that map into the non-coding part of the genome. ATAC-seq has also been implemented in the context of single-cell analysis. There are at least 2 commercial platforms available for this purpose. And importantly, single-cell ATAC-seq has also been combined with single-cell nuclear RNA sequencing to simultaneously assess the transcriptome and epigenome at this level. The last method I wanted to mention, or group of methods, involved the analysis of chromosome conformation capture. As you all know, DNA is folded, chromatin is folded inside cells, in part because the 2 meters of DNA must be fitted within the confined space of the nucleus. In part also because chromatin folding enables interactions between regulatory elements that can be separated by large genomic distances. So this method, it's called chromosome conformation capture analyzed by sequencing, or HiC. It's a kind of prototypical method, and there are some variants of this technique. These techniques involve proximity ligation, cross-linking of the pieces of chromatin that are nearby, then fragmenting the entire genome using restriction enzymes, filling in the overhangs with biotinylated nucleotides, ligating the nearby sequences, and then fragmenting the DNA before immunoprecipitate or streptavidin precipitating the biotinylated products. Then these hybrids are sequenced, and wherever they are enriched, the sequence analysis can identify where in the genome the components of the hybrid sequence came from. This way one can identify the genomic regions that are looping together. There are some important single cell, there are some important methods that are in the development of pipeline that I want to mention. In addition to the ATAC-seq, other epigenomic methods have been implemented at the single cell level. They have not been commercialized yet, but hopefully they will be in the near future. They are available for the analysis of DNA marks, histone marks, transcription factors, even single-cell Hi-C has been published. And quite recently, just last month, a spatial method has been published that permits the analysis of histone marks in a spatial context in situ in tissues. Finally, I want to mention that each field requires standards, and in our field, the ENCODE Consortium, Encyclopedia of DNA Elements Consortium, and NIH-funded consortium takes care of these standards. I would recommend everybody who wants to study epigenetics to go to their website, where you can find experimental guidelines, uniform pipelines for data processing, and quality metrics, which one has to follow in order to publish results nowadays. And I included the link to this website. This is all I had for today in this short talk. Thank you very much for your attention. I'll be happy to answer any questions later in the webinar.
Thanks, Tamas, for a wonderful talk. I'd like to welcome our next speaker, Mayo Kaelberer. She's a sensory neurobiologist. In 2015, she was awarded a PhD from Mayo University in cellular and molecular physiology. At Yale, Mayo studied how individual neurons of the vagus nerve respond to inflammation. Though the vagus has been of interest for centuries, only in recent years have the tools emerged to study single cells. After completing her PhD, Mayo joined the laboratory of Dr. Diego Borges at Duke University. She focused her expertise on uncovering how the gut communicates sensory signals from nutrients to the brain. In 2018, Mayo was the leading author on an article in Science showing the neural basis of the new sense, a gut sense. And more recently, she found that this gut sense can distinguish nutritive sugar from artificial sweetener to drive consumption of nutritive sugar. This work has opened up a new field of exploration in sensory neurobiology, one to explore how nutrients affect emotions and behavior through dedicated neural circuits. So we are pleased to welcome Mayo this evening to talk about the gut sensing toolbox she has developed. Thank you, Mayo.
Thank you so much. Thank you for this very kind invitation to talk about some of the tools that I've been using. So as Andrea very kindly pointed out, my laboratory, we study how it is that the gut communicates to the brain. And our ability to see how it is that the gut communicates to the brain has been pushed forward by the technologies that have been developed, not only for technologies that have been developed in the brain, but how we use those technologies to study how it is that the gut works. And so today I'm going to talk about three tools/techniques that have been key and instrumental in how it is that we have been able to dissect sensory transduction in the gut. So the very first tool I want to talk about is how to explore the circuitry. So in order to establish that there was a connection, the very first thing that we used was a tracing tool. So we used rabies virus in order to trace neural circuitry. And so rabies virus has this ability to cross synapses. So once a neuron is infected, it then crosses the synapse to infect the next neuron. And people have found that this ability to cross the neuron is due to this glycoprotein, this RABG glycoprotein. And so what we do is we use a virus that lacks this glycoprotein. And so then it was only able to spread when that RABG protein is expressed in our cell of interest. In addition, in order to really target the cell that we're interested in, so we are interested in sensory cells of the gut, we use this complementary NDA TVA system where the virus is not only lacking the glycoprotein required for jumping, but is also coated in this envelope A protein that only recognizes the TVA receptor. And so using this virus, we were able to deliver this via enema when it gets into the lumen of the colon, the NV virus recognizes the TVA receptor that is expressed on sensory cells of the colon, which also express the glycoprotein, allowing it to spread one synapse. And so I should say that this experiment, as our GI tract is very good at protecting us from viruses, there are a couple of key features that we had to adapt in order to get this to work in the gut. The first is that it's a very high concentration of virus. So we need to use a titer that's around 10 to the 13th. The second is that the gut is very good at protecting the body from pathogens. And therefore, this has to happen when the mice are very young, between P1 and P3. But when we do this, what we can find is that here we have in red a antibody for PYY, which is the staining of colonic sensory cells. And in green, our virus, in green is our rabies virus. So the rabies virus also drives green fluorescent protein. What we can see here in the 3D reconstruction is that that virus was able to infect that colonic cell. And then since that cell had the glycoprotein, was able to jump onto that fiber. And so very early on in my postdoc, I was able to get this image, which is one of my most favorite videos. So these 3 vagal nodose neurons are directly connected, monosynaptically connected to sensory cells in the colon. And so this allowed us to see that there's actually this very direct connectivity or structure or circuit that is connecting these sensory cells to the brain. And I've taken this technique one step further and made a multisynaptic rabies tracing virus. And so rabies in general is a retrograde virus, meaning it travels in the opposite direction of the activity of communication. However, in the peripheral neurons, it is actually bidirectional. And we think that this is because rabies is utilizing external transport, but these sensory neurons have a special pseudo-unipolar phenotype, which means they are all axons and no dendrites. And so I wanted to know, well, how can I map efferent connectivity down from the brain into the gut? And so instead of expressing just the GFP, I also express this CRE protein in the rabies virus. So now in a wild type mouse, the virus gets into these epithelial cells, but it can't travel any further because there's no glycoprotein. However, in a CRE-dependent rabies G mouse, when the virus infects the cell, since it's also expressing CRE, it drives the expression of rabies G and therefore is able to jump onto the next neuron. And we could see that the nodose ganglia gets very well labeled when I deliver this rabies via gavage. But importantly, when we get into the brain, our rabies is now traveling in a retrograde fashion. So we can find these areas that are now efferently projecting down into the gut. And by timing out the days, since this virus just continues to jump, I'm able to tell what are the secondary, third, fourth, fifth synapses that have an influence on the gut. And so this is just the first tool that we use to establish the basic circuitry of these sensory cells, or neuropod cells, as we call them, that are synaptically connected to, in this case, the vagus nerve is where we focus. The second technique we developed in order to establish what is the communication between these vagal neurons and the neuropod cells. And so in our laboratory, we use this co-culture system where we pull out neuropod cells from the epithelium and we back sort them based on fluorescence, and we culture them with sensory neurons. So here we see in green is our epithelial neuropod cell, and then kind of in red over here, these are the neurons. And what we see is over time, when we culture them together, the cells kind of move around a little bit, the neuropod cell reaches out, and then the neuron connects to it. And this forms this connection in an in vitro culture system of just these 2 cell types. And this allows us to then evaluate how it is that these cell types are communicating with each other. So using this co-culture system, what I did is express a channel rhodopsin in the epithelial cell so I could light activate this. And then I used wild type neurons of the nodos ganglia, the vagal ganglia. And I can record now from these nodos neurons while I give a light stimulus. And when I do this, I see that there's this robust excitatory postsynaptic current in the connected vagal neuron when a light stimulus is applied. And so using this system, we wanted to interrogate what is the actual neurotransmitter that's being released from the neuropod cells and acting on the vagus neuron that allows us to get that excitatory postsynaptic current. So using this system, what we found is that if we block glutamate receptors using kyaneric acid, this is an ionotropic glutamate receptor blocker, we're able to completely abolish that excitatory EPSC. And when we wash away the drug, it comes back, indicating that glutamate is the neurotransmitter that is in this synapse. And that it's one of the neurotransmitters, at least, that's allowing the neuropod cell to communicate with vagal neurons. And so the third tool that I would like to talk about is then how do we get this to behavior? So we have now the circuitry, we know what the structure is. We have a little bit of knowledge about how the communication is taking place, like what are the neurotransmitters, but what does this mean for behavior? And this has been a struggle for us because optogenetics has been a very powerful tool in the brain. However, optogenetics in the gut has been very difficult. And the reason for this is that the stiff fibers will puncture the gut, and it's just hard to find a way to get the light in there. And so we were fortunate to have met Polina Anakiva, who's she's at MIT, and she had developed this flexible fiber optic cable for delivery of light stimuli. And there are some key attributes of this fiber. The first is that this fiber can bend up to 270 degrees and still transmit light. So here, what we have is percent transmission. 100% is the transmission when the fiber is straight. In blue, this is a 90-degree angle. The red line indicates 180 and then a 270-degree turn. And we can see that over these well, let's hope your gut is not making a 270 turn. But over these very large curvatures, this light or this cable is still able to transmit light. The second thing is that the gut is constantly moving, and we also needed a fiber that over constant movement back and forth, it was still maintaining its transmission. And so here now we are saying 100% transmission is the first straight transmission. And then we go through 180 cycle bends to straight, and we can see that it decreases a little bit over time, but it's more or less very, very consistent, indicating that this fiber can take a lot of wear and tear. And so here we have this ultimate test. On the left, we have a stiff silica fiber. So these are generally the fibers that people use when they're using brain optogenetics. You can just anchor it to the brain and it sticks in there. And then on the right, we have our flexible fiber. And what we can see, and then we're holding this over a 1.5 agarose piece of protein. And this is about the stiffness of the gut. And what we see is that silica fiber will penetrate it completely. And we actually tried this, those poor mice. We did try the stiff fiber, and we did find that this is true in the mouse, but the flexible fiber doesn't affect it. A second thing that is very key about this fiber is that you'll notice that the fiber itself, the stiff fiber, there's a point of light at the tip. This is key in the brain because you have one area that you want to excite, whereas this flexible fiber is illuminated across the entire length of the fiber, which for us is a key necessity for any fiber that we use in the gut, because the cells that we are trying to excite are very sparse. And so instead of just exciting a single point, we need to excite a small area. And so we use this flexible fiber in order to then, we expose about 1.5 centimeters, and then we implant it into the gut. So it's now just in the proximal duodenum. And we anchor it to the head because these mice will pull anything out if you put it inside of them. And we anchor it to the head, and now we can actually deliver optogenetic control to a specific cell set within the living and moving animal. So one of the first things that we wanted to do is just test the functionality of this fiber. Is it doing what we think it's doing? So the first is that we did a well-known lipid-induced behavior. And so what is known is that if you give a gavage of intralipid, what you'll see is that the mouse won't eat. And this is a this is a CCK mediated response. And so this is on the left. But if we block CCK cells using halorhodopsin, so this is now with the flexible fiber on the right, what we see is that lipid gavage no longer induces that decrease in consumption, indicating that the fiber is able to block the activity of these CCK cells within the small intestine. And then we're interested in sugar. And so the next question is, if we just use our normal vagal cuff assay that we generally use to test different nutrients in the gut, is the fiber able to recapitulate the same response that we were seeing with the stiff fiber when we put that in the gut? And this is not a survival surgery. And in fact, we do see that this is true. So we see in brown reddish rust color is the normal vagal response to sucrose. The 473 laser is our control laser, so we know there's no heat response. And then the 523, this is this implanted flexible fiber optic, completely abolishes the vagal response to sucrose. And we've pushed this out to about 3 months. We still see a good response. So this is a fairly long-term and durable implant that we've created. And so then I just want to show one application that we had with this was looking at the sugar preference assay. So if you give a mouse the option between the nutritive sugar, sucrose, versus the artificial sweetener, sucralose, and you expose this mouse to these two bottles, over time, they will develop very quickly, actually, they will develop a preference for the nutritive sucrose and not the sucralose. And this has been shown by other groups that this is a post-ingestive effect. So if you knock out taste, this still happens. So something in the gut is sensing this. So we wanted to test in neuropod cells, is this the cell that's able to distinguish between these sugars? And so using this flexible fiber optic, we silenced neuropod cells while the mouse is given this option between sucrose and sucralose, and it completely abolishes their preference. And so it looks like everything went down, but in actuality, these mice are drinking the exact same amount of solution. The only difference is that their cells are no longer able to tell the difference. Their neuropod cells in their gut and proximal duodenum are no longer able to tell the difference. And they now drink equally from these two solutions that are more or less iso-sweet. And so this is just one of these tools that we're now able to use optogenetics in the gut in order to assess a behavior or a consumption behavior in the whole animal. And so in this short amount of time, this is just a quick overview of this gut-brain toolbox as we're trying to create new tools in different ways in order to interrogate a very difficult system, which is the gut. It was so long and it's so complex and mobile all the time that it's been a lot of fun to try and wrap my head around some of these problems. And then with that, I just want to quickly say thanks to my laboratory as well as the Borka's laboratory, and of course my collaborators and support. Thank you.
Great. Thank you, Maya, for a fabulous talk. Well, it's a pleasure to introduce our last speaker this evening, Dr. Brian Gulbransen, who's Associate Professor and MUSU Foundation Professor in the Department of Physiology and Neuroscience program at Michigan State University. Brian received his bachelor's degree in zoology and physiology from the University of Wyoming, where he began studying the enteric nervous system under the mentorship of Dr. Paul Wade. He then completed his doctoral training in neuroscience at the University of Colorado Medical Campus, where he studied interactions between solitary chemoreceptor cells and nerve fibers in the nasal cavity under the mentorship of Dr. Thomas Finger. He then went on to complete postdoc training with Keith Sharkey at the Brain Institute and University of Calgary, where he studied communication between enteric glia and neurons and mechanisms of neurodegeneration in the gut. So Brian's research program focuses on mechanisms of neuroinflammation in the intestine with a specific interest in the role of enteric glia in health and disease. Work from his lab has uncovered some of the fundamental rules that govern intercellular communication between glia, neurons, and immune cells in the gut. He is currently supported by grants from the NIDDK, which focus on the roles of enteric glia and gut motility and pain, and a PPG from NHLBI that focuses on interactions between adipocytes and nerve fibers and perivascular adipose tissue. The focus of his talk this evening will be latest imaging techniques. Brian?
All right, well, thank you very much for that introduction. And thanks again to the ANMS for sponsoring this, this symposium. I think these online sessions are great. And it's really my honor to contribute to the discussion tonight. So my task tonight is to give a quick overview introduction to some of the imaging technologies for neurogastroenterology. And so as there are many of these, I know I'm not going to get to cover everything, but this will be a kind of a brief whirlwind overview of some of these technologies. So the great thing about working in neurogastroenterology is this system really lends itself to imaging. It's a beautiful system to work with from the gut organ level to the cells to the whole gut-brain axis. And the system is just laid out beautifully for multiple different types of imaging. So I think we're really lucky in this field to have this system to work with because it is really amenable to many different types of imaging technologies. So if we did want to image the gut-brain axis, how would we do this? Well, since this is an axis between the gut and the brain, we need tools where we can image at the whole animal level. We have to be able to image things in the gut. We have to be able to image things in the brain and the whole connected system. We have to get down a little bit more specifically at the organ level. So we need tools where we can study the gut itself or the brain. And we need tools where we can look at multicellular networks. So when studying the enteric nervous system in particular, we need to be able to study connectivity of neural networks, how these cells work to coordinate gut functions. And to understand the properties of individual cells, we need cellular imaging resolution. And then to get more granular, we'll also need tools where we can understand subcellular mechanisms and subcellular signaling domains in these cells, within the networks, within the organ, within the animal. So what do we want to think about before we get started with imaging? Well, some of the considerations that we should think about before even starting experiments are, one, what do you want to measure? Because that will tell you pretty quickly if there are reporters available or how you could measure this with imaging. So next, are there reporters available to measure what you want to image, right? And then how will you express these reporters in the cells of interest if they are available? Are there potential complications that you might run into? So movement is a real issue in the gut because it's a moving organ. Diffraction is also an issue and access is an issue in the gut. So these are all considerations to consider when imaging. Specificity is an issue. You know, is whatever driver you're going to use to get your marker or protein, whatever you're going to use to image, is it specific for your cells of interest? And then last, how are you going to analyze this data? And there are multiple ways to do this. So I'll give a quick overview of some of this stuff today. So, okay, so you've decided you want to image, and I just want to give a quick overview of some of the reporter systems that might be available to image some different things. So many of these that we're familiar with are some of the fluorescent probes, and these can be stable probes like this TD tomato image shown here, where we've expressed it in enteric neurons. Switchable reporters, and so these reporters have inverted sequences where they can switch from red to green in the presence of Cree. And this is one example a DREAD system. So it's a chemogenetic system. So in cells where it's expressed in all cells, but only in cells expressing in the presence of Cre, it turns red. And so this is a switchable system. There are also a number of genetically encoded calcium indicators, abbreviated here as GECIs. And so you can express many different versions of these genetically encoded calcium indicators to study calcium activity in various cells or various organelles. You can also use fluorescent probes to study neurotransmitters. And there are these fluorescent false neurotransmitters that can get packaged into vesicles, and you can study neurotransmitter dynamics. Now, there are also many different ways to measure genes. There are voltage-gated sensors and many more. So this is really just a small sampling of the number of different reporter systems. Okay, so now we've picked our reporter and we've decided we want to image. So one of the first levels of imaging we could do would be in vivo imaging. So if you express your reporters in your cells of interest, then you can use a system such as the IVUS system that I'm showing this example here from Chris Contag's group here at MSU. And Chris Contag is one of the developers of the IVUS system. And what this is, it's a non-invasive system that allows you to do longitudinal monitoring in vivo. And so this is pretty useful because you can look at things like cell trafficking, gene expression patterns, and then it also has spatial unmixing capabilities. So you can look at multiple probes at once. And so in this example here, this mouse is expressing a reporter in certain cells, and you can see basically the intensity scale here that's quantitative data related to expression. This is another nice example here shown on the right, where it's showing in vivo monitoring of fluorescent tagged neural crest cells. And so you can watch where these things go in vivo. So another example of in vivo imaging, which wouldn't be in a live animal, is this system. And this is a cryofluorescence tomography system from Emit Imaging. And what this basically does is you can take an experimental animal like a mouse, you can freeze it in a mold, and then basically it's sectioning this and taking an image every 10 microns through the section. So once you get through this entire animal, you can reconstruct the animal. So it's really an ideal system to look at expression of fluorescent markers or proteins through an entire animal. So in this example shown here on the right, this is looking at a fluorescent tagged marker of cancer cells. So using a diagnostic tool to look at where these cancer cells go. Okay, so one of the tools that many of us anyway are familiar with imaging is the confocal microscope. And so this is a traditional benchtop confocal microscope, but you can also miniaturize this to be able to use in vivo, both in experimental animals and in humans. And so this is also a development from Chris Contag's group here at the Institute for Quantitative Health and Engineering at MSU. And so it allows you to do optical sectioning in patients and animal models. And so this is an example of this where they've used one of these confocal microscopes. And so they're doing confocal laser endoscopy here. And so this is a circumferential Raman endoscope. And so this has a rotating head on it that's basically a traditional confocal here that's scanning around in the lumen of the gut. And then you can multiplex this with Raman imaging. And so this is looking at the spectral properties of different chemicals and different proteins. And then you can also enhance this using basically this spatially enhanced Raman scattering nanoparticles. And so this is an example showing some of these nanoparticles. And so if you incorporate these nanoparticles with the traditional Raman scattering, you can even get better signal to noise ratio. So you can also use the same types of techniques in animal models. And so this is the same system, but just miniaturized and put in a backpack on a rat. And so this is the whole confocal system on a rat. And this would be the scanning confocal head that then you can image in vivo and a live moving animal as you're doing experiments. So it's a nice system where you can go anywhere from in humans to animal models. Okay, so let's get a little bit more focused then on the gut itself and organ imaging. So one of the nice technologies that's emerged recently is called light sheet imaging. And this is a really nice technique where you use basically two confocals, one where you shine one plane in through the tissue. And so you just illuminate in one plane and you image perpendicular to that plane. So it's especially useful for very thick things, and it's especially useful for optically cleared things. So this is a technique that's been incorporated in brain imaging. So when doing optical clearing in the brain and then imaging through the whole brain, and it's nice because you get this illumination in one plane, so you don't get as much photo bleaching and imaging effects. So you can increase your signal to noise ratio. And these are a couple of examples of where this has been used in the gut. This is some data on the top right here of a zebrafish gut from Weil and Marion Bronner. And it shows really nicely, you can see the architecture of the enteric nervous system in a piece of zebrafish gut. This has also been done in pieces of human gut, in this example shown down on the bottom right. So you can get the same type of images, both from zebrafish, rodents, all the way up to human gut, where you can see the entire intestine. Okay, so within the intestine, we know we have multicellular networks, and there are many tools to be able to understand, the imaging tools to understand these networks. You can use these stable fluorescent proteins like the TD tomato image shown here on the left to express fluorescent markers in your various cells of interest. Or you can use genetically encoded calcium indicators and other indicators of cellular activity, like this example shown here on the right, where we've expressed genetically encoded calcium indicators in enteric neurons and glia. So this image here is taken this recording is taken from wide field microscopy. And wide field microscopy works pretty well in the enteric nervous system, since it's basically one cell layer thick. But you can really enhance this a lot if you jump up to using confocal imaging and using a resonance scanning confocal microscope. And so this is an example of a resonance scanning confocal microscope from Nikon, where we obtained this recording. And so this is the same animal model as that last recording that I showed you. The difference here is that this is taken at a much faster speed and it's much higher resolution. And so with using a system like this, you can get up to scan rates of about 720 frames per second. And you can do very high resolution imaging to up to 8K in some of these images, but that takes much longer. So another feature of these systems in these confocal microscopes is that they have spectral unmixing. And so this is a nice tool because it allows you to look at multiple different fluorophores and different cell types and separate those signals. So this is basically the same idea as doing flow cytometry, but doing flow cytometry in tissue. And so with the spectral unmixing, you can use a white light laser and a spectral detector to separate emission spectra of fluorescent probes that are very similar in nature to one another. And so this is a nice example that was published by Zeiss, but it's showing a section of gut here labeled with multiple different probes showing multiple different types of cells in the wall of the intestine. And so this is great if you want to look at, different types of immune cells, different markers of multiple cell types in the gut. So another variant of this, but it is a little bit different, is this technique called fluorescence lifetime imaging. And so fluorescence lifetime imaging also allows you to distinguish between fluorophores with similar spectra. And it also allows you to distinguish between molecular environments surrounding the same fluorophore. And basically what you're doing is delivering a fast pulse and then measuring the decay kinetics of the fluorescence of that signal. And so in this example that I'm showing you here that was published in Redox Biology back in 2020, they were looking at these organoids, and they were looking at the basically the oxidation properties within these organoids. And so when this was oxidized, they basically emit with a different decay kinetic based on whether they're oxidized or reduced. And so you can pick that out just with doing fluorescence lifetime imaging. And so you can also do similar things as spectral unmixing with fluorescence lifetime imaging. Okay, so one of the last things, so now we've gotten down even more granular and talk about subcellular events. And so this is another variant basically that I'm showing in these recordings here. And so this is also using these genetically encoded calcium indicators, but here we've expressed them in the cell membrane instead of having these be cytosolic. And what this gives you the ability to do is target these detector proteins to different organelles. And so you can do similar targeting for the endoplasmic reticulum, mitochondria, lots of different places. With this particular model, by targeting the genetically encoded calcium indicator to the cell membrane, you're able to detect near membrane calcium events that don't involve as much summation. So you can see faster events. You can see events where the cytosol is not readily present, like in small glial processes or very small nerve fibers. So there's a lot of techniques like this where you can get down into subcellular ranges. So finally, I just want to say a word about the analysis. So analysis is one of the most challenging aspects of imaging, and there are as many ways to do the analysis as there are imaging. The good thing is that I think during COVID, it's one of the things that people did was develop a lot of analysis programs, and so these are free to the community to use. So I'm showing just naming a few examples of analysis software packages to analyze calcium imaging data. And so many of these are Fiji-based. These are free. One that I wanted to highlight here that I'm showing examples of, this is called STMap Auto. And this is from Sal Baker's group in Reno. And this is a really nice method that they developed. It's all Fiji-based and free. But you can basically get a lot of data out of your calcium imaging where it's going pixel by pixel. And so you can see event amplitude, event duration, propagation, the spatial spread of calcium signals. So it's a good way to analyze single cell events and get a lot of data out of those. But there are many programs where you can kind of see what you need to analyze your data in the best way. And this kind of aspect of imaging could be many seminars put together. So I just want to stop there by thanking the ANMS, thanking my great lab who does great work, and my funding from the National Institutes of Health that support us to do the work that we do. And thank you all for attending today, and I'd be happy to answer any questions.
Great. Thanks, Brian. It's amazing imaging. And I'd just like to thank all our three speakers for today, 3 wonderful talks. We have a short amount of time for questions. If anyone's got any direct questions, to put them into the Q&A box. If you come up with some questions afterwards and you want to continue the conversation with our speakers, you can put them into the doc matter, and there'll be some information coming up on that soon. And that's open to all ANMS members, so you can continue questions and responses in that forum. There are no Q&A, there's no questions in the Q&A, but I do have a quick question for Mayo. It's an amazing toolbox that you're developing. I just want to, I might have missed it in the nutrient sensing experiments, where you inhibited the connection between the neuropods and the sensory endings, and then they could no longer detect the difference between the 2 different nutrients. What do you, I might have missed it in terms of the genetic makeup of the animals. Are you inhibiting, what are you inhibiting, the neuropods or the sensory endings?
Neuropods.
The neuropods.
We expressed the inhibitory halorhodopsin in the neuropod cells.
Okay, I missed that bit, yeah.
Yeah, it's just the epithelial layer that we were blocking.
Yeah, okay. And that was my one. I have other questions, but if there's any others coming up.
Yeah. let me ask a quick question, Tamas, about epigenomics and where we are. I mean, it's very fascinating work. How far away do you think are we from using epigenomics in like personalized medicine in like undefined motility disorders? I mean, it's, where we can actually manipulate RNA and DNA to actually therapeutically treat some of these disorders.
So I think in general, it is still a challenge to use epigenomics in personalized medicine. In a way, it is a challenge to use DNA information in personalized medicine. So it's not surprising that a more complex concept and more complex regulatory mechanism has not been directly implemented in patient care. Nevertheless, there are investigations underway to identify biomarkers and mechanisms that could identify diseases. Probably the most promising aspect of epigenomics is to identify the function of non-coding genetic variants. 92% of all disease-relevant genetic variants are in the non-coding part of the genome. Many are at splice sites, others are intergenic regions, and the only way to tell whether these are functionally significant is by using epigenomic technologies. GTEx consortium published a set of papers, I think it was in 2020, if I remember correctly, 21, on this, and certainly there are some marquee publications already on this subject.
Great, thank you.
Okay. Thank you. Just there's one question in the Q&A, just a quick question, quick answer from Shanthi. This is for Brian. Thank you for sharing all the amazing imaging techniques. In the imaging you mentioned with the colonoscope, in the human colonoscopy data, can it be compared quantitatively?
Yeah, hey, Shanthi. So my understanding is that it can be. So it expands what you can do in a colonoscopy quite a bit. And so they're using this technique, especially to diagnose cancer. And so they're using this also to look at, different markers of disease, I think, in the colon too. But my understanding is that Chris Contag's lab is doing this both clinically and experimentally, and is doing quantitative imaging with this.
That's great. Thank you. Now we're going to have to wrap it up. Thanks for the wonderful talks today, and thanks for everyone joining in the session. We have a few other questions which we can continue those conversations, and we'll post them on Doc Matter, which the URL is coming up now, you see on the screen. So we'd just like to thank everyone for joining this evening's webinar, and to ANMS for organising such amazing virtual abilities to network and see all this amazing technology. Thank you.
Thanks, everybody.